Hello everyone, my name is Paulin. I'm a postdoc associate at Fritz Sedlazeck lab at Baylor College of Medicine, and today I will talk about somatic analysis of cancer samples with TumorLens. So first of all, we want to know why does it matter? So why is it so important? So in personalized medicine in cancer, we know that not all cancers are the same. Two individuals may have the same type of cancer, let's say lung cancer, and maybe even the driver mutation, so the mutation that starts everything, it's the same. But the following mutations will not be the same. So these somatic mutations for each individual will be different. Also, each individual has its own baggage of mutations that come from germline. So also it helps to improve prognosis with personalized medicine. One example is that the detection of loss of HLA genes and other important oncogenes can aid with the prognosis. And also we want to better understand the genetic and epigenetic variations of the tumor. So let's start from the beginning. What are somatic mutations? So these are mutations that occur in the DNA of an organism that are not inherited, meaning that they occur in the cells that are not from germline and they are not from their parents. In these, here we see 4 brains, one has a square. So this you see that everything is red. So this is a germline mutation. It's present in every part of the tissue. And the other 3 examples are somatic mutations, which could be present in partially almost all the tissue, as you see on the top right corner, but all can be also for a specific part of the tissue or specific part of the organ, as you can see in the 2 bottoms. And also many of these mutations are neutral, meaning they don't contribute too much. Some of them are linked to cancer and other diseases. So in cancer specific, so all cancers carry somatic mutations. So the driver mutations that I mentioned at the beginning are a subset of these, and they confer selective growth advantages to the tumor. So they can kind of make the cancer go big, grow, and they're implicated in the cancer development. More than often, these mutations are in DNA repair systems, so this elevates susceptibility to various types of cancers. On the right side, we see kind of the schematic. So on the yellow, we see like normal cells that have germline variants that come from that are inherited, and the black ones are tumor, which have not only the germline variants, but they will have also specific variants that are only detected in the tumor. And these somatic mutations occur in different types. Ones are single nucleotide variants, you can see on the top, then structural variants, you will see an example in the middle, and then copy number variants, you'll see at the bottom. For the single nucleotide variants, they are, as the name say, changes in a single base or nucleotide position in the genome, and if they occur in a gene, they can have an effect. For example, they can be synonymous if the codon doesn't change. They can be non-synonymous if the codon changes, as you see in the middle panel, or they can gain a stop codon, meaning you will have a truncated protein, as you see in the bottom panel. Also, these SNVs aid us to detect loss of heterozygosity, which is a genetic abnormality of diploid organisms in which one of the copies of the gene or part of the genome are lost. So we are diploid organisms. We have inherited one copy from mom, one copy from dad. So if we lose one of those, we see there's loss of the heterozygosity. So these type of kind of abnormality are recurrent in cancer development and can be of 2 different types. One is you have a simple deletion, you miss one of the copies, so you have copy loss LOH. Or after this deletion, you can have a duplication of the remaining allele, so you have 2 copies again, but they are the same, and we will call these copy-neutral loss of heterozygosity. And there are certain tumor types in which loss of heterozygosity can affect substantial portions of the genome. We have developed Spectre, a tool that not only detects copy number variation, but also loss of heterozygosity. Next are structural variants, which are genomic alterations of 50 base pairs and larger, and they are in 5 main categories: deletions, insertions, duplications, inversions, and translocations, and in many cases, combinations of 2 or more of these types. They cause a higher variety or higher diversity when we compare them to single nucleotide variants, just by the sheer amount of bases that they affect. In the lab, we have developed Sniffles2, which is a structural variant detection software. So up to now, in many studies, researchers have mainly focused on copy number variations and single nucleotide variants. However, structural variants play a huge role in cancer. Here's an example of a breast cancer cell in which what you see here are all the connections that occur between like translocations when you jump from one chromosome to another, or inversions when you have jumps from the same chromosome to itself, but that the piece of DNA will burn. And as you can see here, we have so many of these type of events that are like, if you only study single nucleotide variants, then you will miss all of these events which are important. Lastly, we have copy number variations. And these are special types of structural variants because they are duplications or deletions, and they occur when sections of the genome are repeated or missed. The number of repeats varies between individuals, and they can be neutral. For example, they do not much. Among neurons in the human brain, somatic CNVs are frequent, but they are also linked to diseases. Here, I'm showing you one of such examples. So this is from the MECP2 duplication syndrome, which is a neurodevelopmental disorder that is caused by a copy gain in the MECP2 gene on chromosome X. This affects mainly males, as females have 2 copies already and one silenced, but males you only have 1 copy, and then having a second copy causes this disease. So here you see on the top the figure chromosome X, and then located in red the position where this particular gene is, and in the bottom what we see is the coverage pattern of the region. So black and the 0 means that there's no copy number gain. 1 will be like a copy number gain, like a +1, and so on and so forth. So as you can see here, overlapping the MECP2 gene locus, you see like a lot of these red dots, which mean that there is a duplication event, and thus an excess copy of this gene. But not only the differences in the DNA make an effect in cancer. Somatic methylation is also important because in malignant cells are driven not just by genetic changes, but also by epigenetic changes. So the dynamic regulation of DNA methylation is a critical epigenetic mechanism in cancer initiation, maintenance, and progression. So what I mean here is by basically all the mutations I mentioned before, you remove things, you change things. However, here the DNA remains intact. However, the methylation pattern on certain genes or portions of the genome is changed. And thus it can activate or deactivate genes that should not be activated or should be activated. So oncogenic traits can also accumulate through epigenetic disturbances. And the changes in DNA methylation patterns are associated with cancerogenesis and cell proliferation. One of such examples, if you have a gene that aids in cell proliferation, should be most of the cases turned off because the cell should just stay kind of in its own organ. If methylation changes, make that these genes that help cell proliferation start to be transcribed and then translated, then you will have these cells, these cancerogenic cells that will like to grow. And what it has been observed is that hypomethylation can be found genome-wide in some cancers. Finally, we are interested in a particular gene locus, which is the human leukocyte antigen genes, which is a cluster that is in chromosome 6. They encode for cell surface proteins from the immune system. They are highly polymorphic, and they are important for adaptive immune system. For cancer specific, they are important during immunotherapy. For example, loss of HLA genes are important for immunotherapy response, and certain types of HLA genes and types, for example, HLA-A03, benefit less from immunotherapy than if you have all type of HLA-A. So on this particular case, let's kind of go over everything. So the tumors want to escape the immune response. And how they do it, they can do it by the loss of HLA expression. Again, if we go for somatic deletions and mutations, we can have a hard loss, so something that is irreversible, and such as I mentioned, genomic deletion or LOH, or we can have a soft loss, epigenetic modification or methylation that will silence these genes. After these genes are either silenced by epigenetic or genomic deletions, then there's a deregulation of inhibitory proteins, and the tumor can evade the anti-tumor response of the body. So now that we see like what are somatic mutations, and what types of them are, and that they also are interested in methylation. How do we detect them? So in order to detect somatic mutation, we need to distinguish what's germline, so what's from the individual, all the variants are inherited, and what are not, so the somatic variants. So again, in this particular case with the cells that are orange and black, we want to see what all the mutations that have occur in the orange ones that are will be also in the black ones, but we want only to know what are in the black ones. So when we compare between tumor and the patient match control, which also known as normal, we can remove everything that is patient specific and just get what's cancer specific. Up to now, short reads have been fundamental for the detection of somatic variations in cancer. However, they are not enough. So now we are working with long read sequencing, DNA sequencing technologies, in which the reads that generated are significantly longer. For example, short read sequencing usually goes up to 250 base pairs. And for long read sequencing, you are talking about kilobase large reads. And in some cases, for example, for Oxford Nanopore technologies, the ultra long reads can span up to 1 MB or more. Not only that, but we can generate both the DNA and the methylation data in a single run, and we roughly get a 30x human genome coverage per run. And this 30x coverage is very good to detect all types of somatic mutations. So here's one example of how we detect somatic mutations with long-read DNA. So on the right side, you see what's called an IGV plot. So IGV is a software that helps us see how the reads are aligned. On the top is a chromosome, then the genomic region, and then the first panel, you'll see like a gray kind of mountain range. That's a coverage pattern for the control sample. And then all the reads that you see here, like these gray rectangles are the reads that are aligned to this specific part of the genome. All the small dots and colors you see are the small nucleotide variants. And on the bottom, you'll see that there's this black line. So that's the tumor cell line. So you can see that the difference between the control and tumor is that there's a big piece of DNA deleted in the tumor. And this particular deletion overlaps with 2 exons of the PTEN gene, which is an oncogene. For this particular cancer cell line, the COLO829, this is a driver mutation. Additionally, this not only long-read sequencing enables not only the detection of somatic genomic changes as seen here on the right side, but also the epigenetic ones. So with that in mind, we developed TumorLens, which leverage ONT read sequencing to detect somatic variation in cancer and the loss of HLA. So TumorLens is a framework, and it does a comprehensive analysis of the tumor normal cell line. So first, we have a genome-wide cancer-specific somatic variation analysis. So we do SNP, structural variants, copy number variants, LOH, and phasing. So phasing, I haven't mentioned it now. So what it basically does is when you sequence it, you will have sequence-based 2 copies of DNA, one from mom, one from dad. And what we do with phasing is try to separate these 2 copies when they exist. If there's LOH, of course, we cannot do that. But when when you have the 2 copies, kind of separate which reads come from each of the parents. Then we perform HLA-specific assay, which is typing. As I mentioned before, HLA-03 was a type. There are many types because these genes are highly polymorphic, so it's important to know which of the types are we dealing with in each individual. And then we do a genome-wide methylation changes analysis by tumor normal methylation profiling. So in the end, we'll have either the hard loss, which is structural variants, CMV, typing, and loss of heterozygosity, or the soft loss based on methylation. I'm going to show you a couple of examples now. So this is an ovarian cancer cell line, which has a somatic duplication of the HLA locus, or what parts the genes on the HLA locus, on top of a loss of heterozygosity genome-wide. So this again is in chromosome 6. It overlaps with the HLA locus, so it's from 26 to 36 million bases. And what we see here in the tumor on the left, there's a higher coverage in the middle, which is like this is the somatic duplication. When we compare it to the control, we detect that this event is somatic. Next, we focus on the variant fraction, meaning for all the SNPs, if they occur in 1 or 2 alleles, And we see that most of them are in a single allele, and you see this in red. So when the binary fraction goes to 1, it means that we have only one of such alleles present. And in the controls, you can see that there are the red ones, but you also have these blue ones, which means that that goes to 50-50, which is the proportion of each allele that you detect. So you can see that in a particular sample, we have two events overlapping each other. One is a copy number variation on top of loss of heterozygosity. And this is possible with TumorLens. So in a single assay, we can detect various overlapping somatic aberrations. And as mentioned before, these loss of heterozygosity can occur genome-wide. So here's again loss of heterozygosity assessment. It's a different coloring scheme. Red will be, again, as before, the ones that have loss of heterozygosity as the binary fraction goes to 100%. On yellow, which is most of them, the heterozygous signal. And in green is when we have the reference signal, so there's no change. When we compare the control to the tumor, we can drastically see how it changes. So for 9 of the chromosome, we see chromosome-wide loss of heterozygosity. And for another 5 chromosomes, we see partial loss of heterozygosity across the whole chromosome. So the TumorLens not only detects these events in specific genes of interest, such as HLA, but also genome-wide. Next, let's talk about methylation. So as mentioned before, there are methylation changes that will occur in the tumor cell line, in the tumor, cancer. And we want to detect what's going on. So here what we're showing is the methylation difference between the tumor on top and the control. So all the red triangles means that the tumor has a higher methylation signal. And on the bottom is that the control has a higher methylation signal, with gray meaning there's not a meaningful difference. As you can see, most of the chromosome, in this case chromosome 3, is marked almost as blue. When we check this genome-wide, we want to see, okay, how much of these changes go into tumor and control. What we observed is that not only have most of the changes go to blue, but there are a couple of them on the red side. So a couple of regions are highly methylated on the tumor, but most of them are highly methylated in the control, which means we have likely hypomethylation in this chromosome or even in the genome. And then we can look for potential regions in which the change is so abrupt that will be regions of interest for further assessment. So up to now, I've talked to you about all the types of somatic variations and all the methylation changes in HLA. However, things are not as simple as it looks, because in cancer research, most of the tumors that you get are not 100% tumor. They have contaminated with the control. So these contaminated samples, according to, for example, here for the Cancer Genome Atlas, they started that composing at least 80% of the tumor nuclei would be kind of considered enough. However, this ratio was later reduced to 60%, which means that if you get 60% of tumor, 40% of the control, you have enough information such that the interpretation can be good. However, in some cases, 50% of tumor content is the norm, and above that can be a luxury. So what we did in TumorLens is that we need to assess for that. We need to take this into consideration. In order to improve and better have the interpretation of copy number variation, loss of heterozygosity, and typing for the cancers that have less than 100% tumor content. For the case of copy number variation, loss of heterozygosity, I'm going to show a couple of examples. In this case, we need to reassess how much the control is contributing to the tumor. And for typing, we do realignment on the typing results to assess some typing imbalance. What I mean with this is, let's say that you have 2 different types of HLA. You assume that if you map the reads back to these types, you will get a 50-50 if you have 2 individuals. However, if I love heterozygosity, then it would just map to one of them. And if you have some contamination, then you will detect an imbalance in which, if the contamination is 80%, you will see like a 8-2 kind of imbalance. Now, for copy number variation loss of heterozygosity, we did a model in which we have here, for example, on the left the tumor, on the right the control. So again, for us heterozygosity, all the SNPs going close to 100%, meaning that you have like this fixated, so you don't have heterozygosity. After that line of the two, the blue and red ones, you will see like everything looks kind of random. That's what we expect to see. And the control, everything looks kind of random. So this is good. Now, if you have contamination, it means that you will get pieces of the control into the tumor. And we need to model that. How much is the control contributing to the tumor, and how do we detect this contribution? So this is a toy model. Let's assume that my threshold for detecting loss of heterozygosities is having a binary fraction of 0.75, 75%. And I have 100% tumor content, so everything is contributed from the tumor. My threshold still remains 0.75. However, let's say we have a 10% contamination. It means that my threshold would change because the tumor is giving me 9%, but the control is giving me what we assume is like the 50%, because all of this random, it averages to 50%. And the new threshold is 0.725. If we continue doing this up to 50%, you can see that the threshold keeps going low, low, and low. And there will be a point in which the signal-to-noise ratio is going to be low, and we cannot detect anything anymore. So we wanted to investigate how much contamination can we handle. So here are again a couple of examples of the tumor control I showed you before. And now we contaminated this tumor sample with a control at 10, 30, and 50%. And you can see that the red dots are those used for the analysis, how the thresholds go low and low. And then we are still able to detect loss of heterozygosity in this genomic region. Similarly, for copy number variation, in this case, the contribution will be the coverage. So we'll have a 4x coverage here, as mentioned before, and the control has a 2x. And when we do the same type of analysis for the control contribution, we see that if the tumor has contamination of 10%, then your coverage dropped to 3.8, which is close to 4, so it can be rounded up. Already at 30% contamination, the coverage goes to 3.4. And if you have 50% tumor content only, then you reduced from a 4x to 3x. This is important because if the gene is affected not only by, it's kind of dosage dependent, then 4x and 3x has a meaningful difference. And having the knowledge that we have 50% tumor content, we can say, okay, if I've got a 3x, this is likely to have a 4x. So the interpretation can improve. So with all of this, we assessed TumorLens with known samples from benchmarks and cell lines, such as this example. Again, this is also a cell line that we mixed. But we did the analysis of long matched tissue cancer samples. In this case, we have 13 matched tissue samples. And this is ideal for methylation because each cell type has a different methylation pattern. And if you have different cell types, for example, if you control its blood, as many times are, then you don't know if the methylation changes are due to the cancer or due to the cell type. But for here, as we have matched tissues, we know that differences will be based on the cancer and not the tissues. Tumor content are close to 73%. We have one sample at 90, but a couple of 50s. And the coverage, both tumor and control, almost identical, averaging on 28.5x. On these samples, we detected somatic variations. On the, like I have here along 1 to D, and then you can see the number of large somatic deletions duplications. So these are over 1 MB. Large inversions also over 1 MB, the 1 megabase. Somatic loss of heterozygosity, how many regions we detected, all the somatic structural variants and copy number variants, and the number of genes that have methylation differences. This is based on a list of close to 80 genes that we're interested in. But the user can input their own gene list and then will have its own results. So let me show you a couple of examples. Here are large genomic rearrangements in the sample called log5. So you can see here in red are duplications and green are deletions. black will be neutral, meaning like the ploidy here is 2x. So you can see that there are a lot of large duplications in terms of, for example, 2, 3, 8, 9, 16, and 17, and 22. So large rearrangements, large duplications. And we have a couple of deletions, for example, chromosome 2 and 8. Next, we assess methylation. And we see that in long 8, so in this cancer sample, we have a genome-wide hypomethylation. So again, control is in blue, red tumor is in red, and gray is similar. So it means that blue are all the dots in particular chromosome that are higher methylation on the tumor and the control. And you see like a lot of blue and not much red. However, if you see on those that look similar, we see all these black lines are the identity line, which means that there's no difference, that they are identical, both in tumor and control. And you see that all of the gray dots are above this gray line. Even though we see that's a similar methylation pattern, still it's skewed towards higher methylation in the control. So this is a perfect example of a sample that has what seems to be genome-wide hypomethylation. And the final example kind of mixes both. So here we detected contamination by facing haplotypes in the lung cancer 6. So on top is control, on the bottom is tumor, also an HEV plot. It's a bit thinner the reads because the span that we have here is larger, so it's 12 kb. You can see on the top, like the mountain range is the coverage on the control. Then the reads are kind of separated. This is because, as we said before, we phase them, which means that we recognize which reads come from like parent and like father and mother. In this case, we can see based on the structural variants that these are from the same haplotype, so the same parent. Both have a deletion here marked in red and several insertions marked in purple. So it's 1, 2, and 3. All the red dots mean methylation marks. So red is methylated, and when it's not red, it's not methylated. And we know that this sample has 70% tumor content. So what we see next is that on the second allele, what we see is that one of the sample is contaminated. So on the top, you see the control has a lot of reads, but on the bottom, we just have a few of them. What this means is that as we know this sample has contamination, we have loss of heterozygosity on the tumor, and the reads that come from the pink haplotype comes directly from the control, which means is a contamination. On top of that, what we can also see here, so here's a contamination. And what we can also see is that as we have them by haplotype, we know what methylation marks each haplotype has. And we see here in these green boxes that the methylation patterns are different between tumor and control. On the left box, you see the higher methylation on tumor than control, so more red marks. And on the second, on the right-hand side, the green box, we have higher methylation on the tumor than the control. So these patterns are swapped. So to summarize, TumorLens is able to assess somatic variation from single long-read sequencing experiments. We identify variation in cancer such as structural variants, copy number variation, and loss of heterozygosity, HLA typing. TumorLens is able to assess genome-wide methylation differences between tumor and controls, and we can also assess specific genes if the user is interested. We assess the HLA gene locus and other oncogenes, and we can adjust for contaminated samples up to 50% tumor content. But wait, there's one more thing. What if we don't have a control? And this happens quite often. Either the lab missed the sample, there's no sample. Well, we can do pseudo-somatic variation analysis. In this case, we're using a tool called STIX, which is a population annotation tool. What it does is take your structural variants and tells you how many of those it can detect in a population. In this case, the population is 1,000 genomes, so it's a wide variety of ethnicities. And it can be used to prioritize unknown variants. In this case, we have prioritized that this deletion that we found in this stomach cancer sample, chromosome 9, which has not been detected previously in any of the 1,000 genome sequence from 1,000 genomes. With that, I want to thank everyone from Baylor College of Medicine, everyone from Fritz Sedlazeck lab here last year retreat, and Chris Hammer from which we started this project when he was at Genentech, now at Los Altos, and you for your attention. Thank you.